Steps in the Synthesis and Regulation of Thyroid Hormone by Cassandra Cottrell, Bio 202, Monday, Wednesday, Friday from 7:15 to 8:10. The thyroid gland lies inferior to thyroid cartilage of the larynx. The thyroid has several functions. The thyroid binds to receptors in the cytoplasm on the surfaces of mitochondria and in the nucleus. Thyroid hormones bound to cytoplasmic receptors are essentially held in storage. If extracellular levels of the thyroid hormones decline, the bound thyroid hormones are released into the cytoplasm. The thyroid hormones binding to mitochondria increase the rates of mitochondrial ATP production. The binding receptors in the nucleus activates genes that control the synthesis of enzymes involved in energy transformation and utilization. Thyroid follicles are hollow spheres lined by cuboidal epithelium. Cells surround follicle cavity that contain viscous colloid. Step 1 in the synthesis and regulation of a thyroid hormone include iodine ions are being brought back to the thyroid gland via the bloodstream. Iodide ions are absorbed from the diet at the digestive tract. Thyroid-stimulating hormones, sensitive carrier proteins in the basement membrane of the follicle cells, actively transport iodide ions into the cytoplasm. Step 2 in the synthesis and regulation of the thyroid hormone are the iodide ions diffusing to the apical surface of each follicle cell. At the follicle cell, they are converted to an activated form of iodine by the enzyme thyroid peroxidase. This sequence occurs at the apical membrane surface. The sequence also attaches one or two iodide ions to the tyrosine portions of the thyroglobulin molecule within the follicle cavity. Step 3 in the synthesis and regulation of thyroid hormone. Tyrosine molecules with attached iodine ions become linked by covalent bonds, forming molecules of thyroid hormones that remain incorporated into thyroglobulins within the follicle cavity. Thyroid peroxidase carries out the pairing process. The thyroid hormone thyroxine, which is T4, contains 4 iodide ions. T3 contains 3 iodide ions. Eventually, each molecule of thyroglobulin contains 4 to 8 of T3 and or T4 hormones. The major factor controlling the rate of thyroid hormone release is the concentration of thyroid-stimulating hormone in circulating blood. Thyroid-stimulating hormone stimulates iodide transport into the follicle cells and stimulates the production of thyroglobulin and thyroid peroxidase. Thyroid-stimulating hormone also stimulates the release of thyroid hormones. Under the influence of thyroid-stimulating hormones, the following steps occur. Step 4 in the synthesis and regulation of thyroid hormone. Follicle cells remove thyroglobulin from the follicles by endocytosis. Step 5 in the synthesis and regulation of thyroid hormone. Lysosome enzymes break down the thyroglobulin and the amino acid and thyroid hormones enter the cytoplasm. The amino acids are then recycled and used to synthesize more thyroglobulin. Step 6 in the synthesis and regulation of thyroid hormone. The released molecules of T3 and T4 diffuse across the basement membrane and enter the bloodstream. About 90% of all thyroid secretions is T4, and T3 is secreted in smaller amounts. Step 7 in the synthesis and regulation of thyroid hormone. About 75% of the T4 molecules and about 70% of the T3 molecules entering the bloodstream become attached to transport proteins called thyroid-binding globulins. The rest of the T4 and T3 in the circulation is attached to albumin, which is one of the plasma proteins. Only the relatively small quantities of thyroid hormone that remain unbound are free to diffuse into the peripheral tissues. Homeostasis is when there are normal amounts of T3 and T4 concentrations in the blood and normal body temperatures. Homeostasis is disturbed when there is a decrease of T3 and T4 concentrations in the blood or low body temperature. The hypothalamus will release thyroid-releasing hormone. After thyroid-releasing hormone is released, the anterior lobe of the pituitary gland releases thyroid-stimulating hormone. When the thyroid becomes stimulated, the thyroid will then release T3 and T4 hormones. Homeostasis then becomes restored by an increased amount of T3 and T4 concentrations in the blood.